Okay, so I want to tell you that what we're doing right now is being recorded. My voice, your voice, and whatever you see on the screen is being recorded, and then I'm going to post it on YouTube after class. So tonight or tomorrow night, if you want to go back and see, oh, I forgot what she said about cell respiration, or I need to review what she said about mitosis, you can go on YouTube. You search for my name, Patricia Steinke, and then look for our class, Lab Test 2 Review, and it'll be there. And again, like any YouTube video, you can move it forward to whatever parts you want to listen to or review. Okay, so it will be posted. Okay. So I'm going over your review sheet, and this is exercise 5 on enzymes. It says identify reactants, products, and enzymes in a chemical reaction. So I have this chemical reaction, and I picked this chemical reaction because it was the chemical reaction that you and I did together in lab when we did enzymes. And so we're supposed to figure out what the reactants are on this chemical reaction. Anybody tell me what the reactants are for this chemical reaction? Hydrogen peroxide. So I'm just going to put an R there for reactants. How about products? H2O and oxygen. So I'm just putting P for products. And then I added the word enzymes in my question. Is there an enzyme up here? Yes. Catalase. And I'm just going to put a lowercase e. Why did I choose lowercase e instead of uppercase e? Uppercase e I reserve for energy. So lowercase e means enzyme. Now, because this reactant, hydrogen peroxide, uses an enzyme, you may also call that a substrate. This is a reactant, and that is true. It's also known as a substrate because it uses an enzyme. So it's just like you're known as student, but Eric, you're also known as Eric. So I could call you student, and I wouldn't be miscalling you Eric. You are. I could say student, and you would say yes. But I can also call you Eric, right? Right. Okay. Video count number 1. Okay, number 2. Nobody's going to actually have to define any of these words, but on page 57 in your lab manual, if you'll join me on page 57, what you are going to have to do is properly label a diagram. So at the bottom of page 57, you can see a diagram. And I have a diagram that is similar but different. I got this one out of, I think, our textbook. I don't know where I found it. But page 57 is similar to this. And see what I did? I crossed out all of the words. See, you can't see the words because Miss Steinke knows about whiteout. Okay? And then you have to tell me what each part is. So most students can figure it out, but I just want to go through 2 things with you very quickly. The difference between an enzyme and a substrate. Most students tell me they can tell an enzyme because of the depressions, and I'm just highlighting these depressions right here. What do you call those depressions? Active site. So most people can spot an enzyme because of its shape when it's depressions, known as the active site. But I do want to clue you into another way you can easily spot the enzyme. The enzyme does not change in the visual. Everybody, look how the enzyme does not change. As the arrows go forward in time, I see the enzyme does not change, but the substrate does. Check out the picture here. The substrate's right here. It's this 4 beaded thing right here, and then it binds to the enzyme and it changes. What does the substrate change into? Product. Substrates change into products. So substrates change, enzymes don't. Okay, so again, most of you can spot it, but in case you can't, enzymes don't change, substrates do. And of course, when they're together, the substrates with the enzyme, we call that the enzyme-substrate complex. So just glance at these words that were listed here. Enzyme-substrate active site and enzyme-substrate complex were the 4 vocabulary terms. Those will be words that you have to be able to put on a picture. Is okay? All right, great. All right, the next part of that question says, what's the effect of an enzyme on the energy of activation of a chemical reaction? Now, guys, I drew a chemical reaction up here. This is an exergonic reaction, an energy-releasing reaction, you may remember, because the energy of the reactants and the energy of the products are what I just indicated here. The energy of the reactants is higher than the energy of the products, so this thing's going to give off energy. Now, one of these lines, the red one, and one of these lines, the yellow one, one's with an enzyme, one's not with an enzyme. Would it be the yellow one or the red one that has the enzyme? The yellow. How did you know? Lower the energy of activation. Yeah. The energy of activation with the enzyme, which is this one, is lower. So the answer to the second part of number 2, what is the effect of an enzyme on the activation energy of a chemical reaction? What's the answer? It's lower. The enzyme lowers the energy of activation. Lowers. It lowers it. Okay, so in summary, an enzyme lowers the energy of activation, thus the reaction goes faster. So that's the beauty of an enzyme. It makes things go faster. Enzymes make things go faster? I guess beats me. Carry on count number 2. Super number 3. What happens when an enzyme is denatured? It doesn't work, can't function anymore. And then what physically happens to it? Changes shape. Good. So I'm going to write that down. It changes shape. So nice. Yeah, I like that. That's great. That's so my generation. You guys, really. It stops working. Are you okay with that? Good. Second part. What conditions might cause an enzyme to denature? Okay, high temperature. I'm going to write down high temperature. And then pH. And I'm going to write extreme pH. Really high pHs or really low pHs. But okay, on question 3. Question 4. What's the function of catalase? Well, we had it a minute ago. In case you don't remember catalase, page 58. I'm going to write that here for you. Page 58 is where you can find catalase and what it does. Page 58. The function of catalase. Anybody? Speed up a reaction, but what reaction does it speed up? The chemical reaction, the breakdown of hydrogen peroxide. So I'll write that down. I'm going to put it all together. Speeds the breakdown of hydrogen peroxide. Turns out aerobic cells need that to happen. Hydrogen peroxide is a waste product of most aerobic cells, and they need to break it down. Second part of question number 4. How did we judge if catalase made the reaction occur? What do we look for? Bubbles. What kind of bubbles? Oxygen. Everybody okay? Super. Number 5. What effects do different temperatures have on catalase activity? Well, will you join me, please, on page 61? And in your lab manual, you probably have data. Probably some data there. Page 61. Test tube number 1 was in the refrigerator. It was cold. Did catalase work really well at cold temperatures? Did it not work well? No bubbles? Lots of bubbles? Look at your data. Page 60. Am I saying 61? I'm sorry, 60. I apologize, Geneva. So what did catalase do in test tube number 1? Good bubbles? No bubbles? What happened? Bubbles? It's okay? Okay, so it's pretty good. It's okay. How about test tube number 2? We had a warm water bath. I think it was 40 degrees. How'd that work out? Good? Yep. I remember both of these were pretty good. It really depended upon the group. Some of you voted that number 1 was your best. Some of you voted that number 2 was your best. How about the hot water, which I think was at 90 degrees? How'd that work out? It was denatured, right. So that's because high temps denature enzymes. And that's true for most all enzymes. Not going to lie, there are some enzymes that work at high temperatures. They are found in organisms that normally live in high temperatures. So there are some algae and some bacteria that can live at high temperatures, like in hot springs. And they have enzymes that can work at high temperatures, but they're the exception. Most enzymes cannot work at high temperatures. But okay, number 5. Number 6 is an omit. We didn't do that activity, so we're going to omit that question. But we did do number 7. Number 7, would you join me? Page 62. This is page 62. In your lab manual, page 62. That's not supposed to be that kind of do. That's supposed to be this do. Well, anyway. For this question, I drew 3 test tubes. And here was the question. What's the effect of a pH outside the optimum range of an enzyme? What will be the expected effect of acid and base on catalase? So I drew our 3 test tubes. And again, this is page 62. In test tube number 1, test tube number 2, and test tube number 3, they each had catalase and hydrogen peroxide. So if you'll notice, I put catalase and hydrogen peroxide, follow the blue arrows, I put those in all 3 test tubes. And then in test tube number 1, I put acid. Test tube number 2, I put water. Test tube number 3, I put base. Now, let us first look at our data table and help me out. What was the pH of test tube number 1? So really low. What was the pH of test tube number 2? 6. Anybody get 7? Yeah. And what was the pH of test tube number 3? 14. Anybody get 13? 14? Dang, that's high. Yeah, some strong stuff. Okay, now let's look at our bubbling data. Did we get bubbling in test tube 1? Test tube 2? Test tube 3? Okay. So, first of all, what was the optimum pH of catalase? Around 7. How did you know? Because it got good bubbling. If you take catalase above its optimum pH or below its optimum pH, it doesn't work very well. Why? Nope, it's denatured. The extra hydrogen ions or the lack of hydrogen ions change its shape. So the enzyme denatures. So at the low pH or the high pH, enzymes denature. They change their shape. So that the enzyme denatured in 1 and the enzyme denatured in 2. 3. But the enzyme worked in test tube 2 because that was the optimum pH. It was the pH it liked the best. So that's why biological systems are buffered. They try and keep their pH at the optimum so that their enzymes work. So if their enzymes get denatured, they stop working, and that's not good for life. We want our enzymes to work. Okay, so look at question 7. Make sure you're good on question 7. Question 7. Okay, now let's look at questions 1 through 7, omit number 6. See if anybody needed anything repeated before we leave the enzyme lab. Okay. So just staying in order with our laboratory manual, let's go to photosynthesis next. Page 67. There's that really great picture that you guys know I'm a big fan of on page 67. And there's also a chemical reaction there. We have to know it. So get me started. What are the reactants of photosynthesis? Water. CO2 plus water. What are the products of photosynthesis? Carbs and oxygen. That's good. Carbohydrate, usually sugar, and then oxygen. Photosynthesis. It's page 67. This is still from page 67. Next part. In what plant organelle does photosynthesis occur? Chloroplast. Good. Okay, on number 1. Number 2 is page 68. So on page 68, we did this really cool process called paper chromatography. What solvent is used when we did paper chromatography? Well, you guys may remember I had you waft it to smell it. It looked like water, but it wasn't. It was acetone and ether. Now, I'm not going to have acetone and ether as a choice. Here's what I want to have. We'll have nonpolar. What's that mean, nonpolar? No charge. And I know you may recall we talked about why I chose a nonpolar solvent. The pigments in a leaf don't dissolve in water. They have to be dissolved in a nonpolar solvent. If you just put water in that test tube, those pigments would have just stayed on that piece of paper and wouldn't have done anything. To get them to move on the paper and separate, we had to have something that could dissolve them. And because they're nonpolar, I chose nonpolar solvents. The pigments are nonpolar, so we used a nonpolar solvent. And you guys are right. It would smell like acetone, fingernail polish remover. It also had ether in it.
Nail polish?
Nail polish.
Nail polish polar?
No, nail polish is nonpolar. So it's the base that they dissolve the pigments in for nail polish is basically acetone and ether.
Okay, so
That's why it dries so fast. Yeah. Or not, just depends. Okay. How does paper chromatography separate pigments? Based on what? Look at the bottom, page 68. Solubility. Good, Eric. It separates pigments, and you already have this on page 68. It separates pigments of the leaf that we did based on solubility, how well they dissolve. So the process was called paper chromatography. It separates pigments, and how it does that is that the pigments have different solubilities. Okay, still on page 68, and I drew a paper chromatogram where the pigments have been separated, and I have the 4 pigments here. Look on page 68. What would be this top pigment that was yellow? That's carotene. That's good. What would be the next pigment that is also yellow? Xanthophyll. What would be the third pigment? It's green. Chlorophyll who? Good. And what would be the last pigment? It's also green. Chlorophyll B. And you'll notice I used just a little bit different color of green because you may remember that A was kind of more of a bluish green and chlorophyll B was more of a brownish green. You could tell the difference between the two. They were two different colors of green. Now if I give you these four choices, make sure you know who's at the top, who comes next, and who's in dead last. Okay? So know them in order. Obviously, carotene was the most soluble and chlorophyll B was the least. But okay with that? Okay, try this question I have at the bottom. Which of those four was the most non-polar? No. Carotene. It was the most non-polar because it dissolved the best in the non-polar solvent and went the farthest. Carotene is the most non-polar. It went the farthest. Okay, look at question number 2. Make sure you're good for question number 2 on photosynthesis. Okay, on our way to the next question, just take a quick glance at page 70. I like page 70 because it shows you that white light is actually composed of all the colors of the rainbow. I like page 71. We did that experiment. Remember we had a really bright light, big beaker of water, and then a flask full of aquatic plant? Yeah, we did that one. Okay, now we're going to go to page 72. And there was a graph there. Study the absorption spectrum on page 72. What color or colors of light does chlorophyll absorb the best? Now let's just look at chlorophyll A because that one's the most abundant and of course most essential pigment of a plant. Chlorophyll A. What color or colors of light does chlorophyll absorb the best? Violet. Anything else? Yeah, orange looks really good. Red, it's pretty good. Orange is really good. What color or colors of light does chlorophyll absorb the least? Green.
Red, blue,
The least?
green.
Yeah, it starts coming down around blue. Yeah. Coming down. Green is good. It's kind of low in yellow, but yellow, it starts to come back up a little there. I'm going to go green. All right, so how did you know which ones to pick for the best? What'd you look for?
Absorption.
You looked at your peaks because the y-axis is absorption. So for high absorption, we looked for peaks. How did we know what it absorbed the least? We looked for the valleys, the low parts. The low parts, because the y-axis was absorption. So if you want to know what it's absorbing the best, you look for high values. If you want to know what absorbs the least, you look for low values. But it's okay on that. Super. Okay, stay on page 72, look at the graph, and you can see that I have chlorophyll A on the graph, chlorophyll B on the graph, and the carotenoids. Now, we had just done that day paper chromatography, and we knew all these pigments were in the leaf. We found all these pigments in the leaf. Question 4. Why is it beneficial to have several different plant pigments involved in photosynthesis?
More light.
What's capital E? Energy. You can absorb more colors of light, you're absorbing more energy. Okay? Still on question 4. Given your answer, what color or colors of light is the best for photosynthesis?
It's like we're repeating.
I'm sorry?
It feels like we're repeating it.
Mm-hmm. You're going violet? Green? Did somebody say that?
Violet is orange. It's blue. It's violet and orange.
Starts with the letter W.
White.
Thank you.
Oh, that's cheating.
Feels like we're repeating. Good thing, okay? Because you forgot.
So,
Why is white the best color of light?
because it has all these other.
Because it has all the colors. So you get your orange, people. You get your violet. Those of you that wanted green, I don't know why, but you get that too. It's all the colors. Okay, what color is the worst light for photosynthesis? Green.
No. Ultraviolet.
Thank you. Of the visible light spectrum.
Just kill it.
You okay? Everybody's fine? Super. True or false? True or false? Most plants carry out photosynthesis only during the day. That's true. Because they have to have light. That's the photo of photosynthesis. They need light. Second part of number 5, true or false? Plants carry out cellular respiration only at night. That's false. When do plants do cell respiration? Always. Thank you, Eric. Always. Every second of every day, all cells are respiring of some type. Plants are aerobically respiring, but all cells respire all the time. Respiration happens all the time in all cells. Okay, on number 5. Number 6. Okay, let me show you a picture. There was this time where we had, I think, flasks, and we put water in the flask, we put phenol red in the flask, we add a little piece of plant to the flask, and what was most important that I think you'll remember is we put a straw in the flask, and you had to blow into the straw. Okay, now I didn't draw a flask, I drew a beaker. This is what it kind of looked like. Turned pink. Now, when you started, the flask was pink, but when you blew with the straw into the flask, the pink turned yellow. If you don't remember why, I believe we wrote it down on page 74. Page 74. So there was water in your flask. You added CO2. What did you make? That's right, Terry. H2CO3. You made acid. And that changed the pH, thus changed the color of the phenol red. So you put water in the flask, then you, with your breath, you blew CO2. And why I really like you guys to see that is so you can see how soluble CO2 is with water. CO2 dissolves very easily into water and makes carbonic acid. It's really easy. And that changes the pH. So CO2 in the air is constantly dissolving in water. Okay. So then we left the flasks either in the window or in a drawer. So this is the one that we put in the window. Why did the solution that was yellow turn back to red after being left in the window for 2 days? So look at your notes. We came back 2 days later, and it was yellow, and now it was kind of a pinkish color. Why?
Photosynthesis.
Yeah. Okay, so photosynthesis will not be an answer. It's 2 third grade. And it has nothing to do with oxygen. How did you change the color? What gas changed the color? Carbon dioxide. So talk to me about carbon dioxide. Thanks for coming, Lewis. Do it again, Linda. Mm-hmm. Okay, so the plant in the window wanted to do photosynthesis, and it needed some CO2. So it took the CO2 out of the water, thus reducing the amount of CO2 in the water. Or you could think of it, it reversed what you did with the straw. You put the CO2 into the water, the plant took it back out, reversing what you had done 2 days prior. Now, don't get me wrong, this plant made oxygen, I agree, but oxygen doesn't change the color of the water. It's CO2 that changes the color of the water. Okay, so this is all about CO2. This experiment was an illustration of CO2. Everybody okay on that? All right, next part. This is my drawing for darkness. See, there's my moon. Okay? When we came back 2 days later, it was still yellow. We left it yellow. We came back 2 days later, it was still yellow. How come?
No sun, no photosynthesis.
Yeah, with no light, no photosynthesis. So the CO2 stayed in the water. So the yellow means there was still plenty of CO2 in the water. The water still had lots of CO2 in it. This is just illustrating the need for light to do photosynthesis. Now, guys, I know you know that, but we want to just prove it to you. We've learned that over time, plants have to have light to do photosynthesis, but I just wanted to show you for sure they couldn't use CO2 and do photosynthesis without light. It's okay on that one. Okay, number 7. Why is it important to have autotrophs, heterotrophs, and decomposers in an ecosystem? Okay, well, I drew one. I drew an ecosystem. It's a little aquarium. It's very good. There's some aquatic plant here. I got 2 goldfish. They're pooping. That one's pooping a lot.
He's pooping on the other fish, though.
Well,
Or fish.
this is a 2-dimensional drawing. Just pretend 3-dimensionally the poop just falls to the bottom. Okay. And then you see the rocks at the bottom? You guys see the rocks? The rocks are important. You've got to have some rocks at the bottom. Does anybody know why? Thank you. Eric's, you're on fire today. You've got to give the bacteria some surface area to grow on. Yeah, because you need the bacteria. So let's talk about, first of all, who is the autotroph in my pretend ecosystem? The plants, thank you. Your people are scaring me. The plants are the autotrophs. Who are the heterotrophs in my pretend ecosystem? The fish. And who are the decomposers in my pretend ecosystem? The bacteria. The bacteria. Bacteria. Okay, so I've got a little system of gases going around here. I don't know if you can tell, but the plant is making, by photosynthesis, oxygen and carbohydrate. Those are the products of photosynthesis. The plant is giving off oxygen, and that's carbohydrate. And the carbohydrate, don't get me wrong, it would be here in the leaf, but the fish would eat the leaf. Okay, do you eat leaves?
Yes.
Me too. Lettuce, spinach. You guys eat leaves? Okay, Josh. Okay, so the fish eats the plant getting carbohydrate, and the fish, using its gills, gets the oxygen out of the water. Okay, then the fish does cellular respiration, giving off CO2 and water. And then the plant needs the CO2 and the water. Does everybody see we got a cycle going on here? If you don't like my drawing, you might like the one on page 74 better. It's a cow and corn. Maybe you like that artwork better. I can't draw a cow. Okay. Now, the plant is going to have some leaves that fall off and die, and they'll fall to the bottom. And obviously, the fish are going to have waste products that will fall to the bottom. What's going to happen to those waste products that fall to the bottom? They're going to decompose. Who will decompose them? The bacteria. What if the bacteria weren't there?
It would smell really bad.
The waste products would build up, and that would eventually make the water toxic for both the fish and the plant. So you can see how important the decomposers are. Yes? Okay. Everybody notice I got Mr. Sunshine in my drawing. They have Mr. Sunshine in the drawing on page 74 too. Why do we need Mr. Sunshine? To do photosynthesis to make this cycle run. We got to have that. All right, one thing I added to my drawing that they don't have in your book is I put a lid on this, and I want to talk about that. If you put a lid on this pretend aquarium, would the aquarium cycle as long as if the lid weren't there? Would you rather put the lid on it or don't put the lid on it? Don't put the lid on it. When you put the lid on it, then you're cutting off the atmosphere so that you can get even more oxygen or more CO2 coming in. Okay, so having the lid on it is not good. Having the lid off is better. And theoretically, you could put the lid on this if you have this balanced perfectly, and it should last. I had a student, and they gave me this glass ball. It had some shrimp and some algae living in it, and it made it for about 6 months. And then the shrimp died. The algae's still doing great, but okay. But yeah, if you leave the lid off, then this should do better for a longer period of time. Yes? But it's okay on why I put the little lid on there, just to get you to think about how the atmosphere helps replenish gases, allows some things to go away and come in. Why would you want to put a lid on this? Is there any benefit to putting a lid on this?
You got tired of your fish?
Well, they may die sooner. The only reason you would want to put a lid on this is if you're tired of putting water in. Because what's going to happen to the water?
I've always
It's
noticed
going to
that.
evaporate. So you're going to have to constantly be putting some water in, kind of watching your water level. That'd be the only reason that if you put a lid on it, that the water will just recycle and you won't have to put water in there.
My little sister said to me a few days ago she had to refill water. She's like, that fish drinks a lot of water.
Yeah, that's what's happening, yeah. Might want to tell her different. Okay, let's move on to cellular respiration, guys. Page 77 and page 78. I'm going to start on page 77 with cellular respiration. Page 77. I have know the reactants and products of aerobic cellular respiration. That's the one that's on page 77. Check out page 77. How do you know that one's aerobic? As it's got oxygen as a reactant. So there's how I know this one's aerobic. Right there. Okay, so the C6H12O6. What do most people call that? Glucose. Interestingly, fructose has the same empirical formula. The atoms are just arranged differently, but fructose is also C6H12O6, which is a real common sugar that you guys drink. Me too.
Are the sixes important in the rest of the formula?
Not for we will have to do that, but they are important. And balancing equations is always important, but they won't be an issue for us for testing purposes. Good question.
It's always
Okay, and then our products are carbon dioxide, water, and energy. You have to know how many ATP per glucose you can get if you do aerobic cellular respiration. It's 38. So I'm going to squeeze that in right here. 38. 38. You have to know that all the way to the final exam. 38.
With aerobic, right?
That's, yes. Okay, now join me on page 78. We did 2 more. One of them is already in our lab manual. Glucose goes to form carbon dioxide, ethanol, and 2 ATP. And then I think we added glucose goes to form lactate or lactic acid and 2 ATP. Did we do that? Did we add that? Yes. Okay, both of these, you tell me, is this aerobic or anaerobic? These are both anaerobic. How do you know? No oxygen as a reactant. The top one makes alcohol, and the bottom one makes lactic acid. Okay, so far on these? 'Cause I have some questions about them. Okay. Question number 2 I like. Both anaerobic, these two, and aerobic ordinarily begin with what molecule? Glucose. If you'll check it out, they both start with glucose. It's the most common fuel that cells use for any kind of respiration. Aerobic or anaerobic, cells like glucose. Okay, question 3. In which, you get all 3 of those choices, in which of those 3 reactions is the glucose completely broken down? If you completely break down glucose, you harvest the most energy out of it. So which one would that be? Very good. We know it's completely broken down because we've harvested the most energy out of it. And of course, the second part of number 3, everybody knows that if you do it aerobically, you get 38 ATP. What if you break down glucose anaerobically? 2. So 38 ATP if you do it aerobically, 2 if you do it anaerobically. It's a huge difference. Okay, I have some just general questions for you going back to these 3 reactions. Okay, so these are on page 78. This one was on page 77. You get these 3 reactions for choices. Here we go. Which of these reactions would be used to make beer? Now this one. This one you'd use to make beer. Or wine, or any kind of liquor. Just, you get different liquors based on the different things you ferment. Is okay on that one? Okay, which one of these 3 reactions are you doing right now? Yeah, you're doing this one. Aren't you using oxygen and making ATP? Yeah.
If I'm really out of shape, holding this pencil, using
Okay, so Benjamin's point is, if we all went to the gym and everybody loaded up a bar with a whole bunch of weight, it'd be like maybe 20 pounds for me, but for you it may be, I don't know, 100 pounds. I don't know what you can do. And we all started bench pressing. Pretty soon our chest would start to burn, okay, for bench pressing. What's going on then? Your muscles start to burn. What are you doing? We're doing lactic acid. Yeah. This is when your muscles burn. You've gone anaerobic, and you've produced lactic acid. Yeah, you're doing that one. Okay, I got another one. Which one of these is used to make bread rise? The first one, yeah, this one. What gas is making the bread rise? You need a gas, people. Yeast is not a gas. Yeast is cells. To make bread rise, you're going to have bubbles. You guys seen bread? There's bubbles in it. A gas made those bubbles. What's the gas that made the bubbles in bread? Carbon dioxide. I agree you got to have the yeast, but the yeast made the gas that made the bread rise. Yeast by itself won't make the bread rise. You got to give yeast sugar, and then it will ferment the sugar and make carbon dioxide, which will make the bread rise. Okay, the yeast also made alcohol. How come you don't get drunk when you eat bread? Because it evaporates away. Yeah, the alcohol evaporates away. Some of you were telling me sometimes you open a loaf of bread that's older, you can kind of smell the alcohol.
Sourdough.
Like sourdough. Yeah. Okay, so you can get beer and bread from that. Okay, so how about this? How do you get sour cream?
It's bacteria.
Bacteria doing which of these three processes?
It's lactating.
It's lactic acid, yeah. It's the sours from an acid. So here's sour cream. Anything pickled is lactic acid. My personal favorite is blue cheese. You know how tart blue cheese is? Got that kind of sour taste? That's lactic acid. Yeah.
Is breastfeeding?
I'm sorry?
Breastfeeding? Is it lactic acid?
Lac refers to milk.
Okay.
Lactose is a 3-carbon sugar, and then with a few atoms changes, it becomes lactic acid. So LAC does refer to milk. That is true. That is true.
Because when a mom's breastfeeding, it's called lactating, right?
Exactly. And the sugar in milk is called lactose. So LAC refers to milk. It truly does. But okay on those just general questions. Super. We call those application questions in case you're like, what's she doing? Did that. Okay, the fermentation experiment. I want to have you guys go over a couple pages. First, we took our data on page 82. And guys, I did set up the fermentation experiment again in some test tubes so you can see, just visually see it. This one had the itty-bitty tubes. We put sugar and yeast in them, and then we had to shove them up in another tube and do the flippy thing. Okay, that was the fermentation experiment. Let's look at our data. Tube 1 was glucose and yeast. Tube 2 was fructose and yeast. Tube 3 was sucrose and yeast. And tube 4 was water and yeast. Now, correct me if I'm wrong. Did glucose get first place? Yes. Fructose got second place. Sucrose got third. Water came in dead last? Okay. Well, obviously, we put sugars into each of these test tubes. What will I ask you? First, you have to know that fermentation is anaerobic. That what they were doing was alcohol fermentation, and it was anaerobic. What does that mean? No oxygen. This was anaerobic. Okay, why did glucose get first place? It's a simple sugar. I'm going to put mono up here. What's mono mean? Single. It's a monosaccharide. Fructose, I think, came in a close second. Is that true? Again, another mono. Why did sucrose get third place? It's a di. So what does the yeast cell have to do before it could use sucrose? Break it apart. Why did water get dead last? There's no sugar there. Why did we have test tube 4? Yeah, this was our negative control.
So you wouldn't use sucrose for bread.
It just takes longer. Yeah, and I think most people do use sucrose for bread because sucrose is table sugar, but it does. But I think you have to let bread rise a good while. Yeah, it just takes them a little longer. Yeah. Okay, next question on this. What was the gas that we were measuring?
Carbon dioxide.
Thank you. CO2 was the gas that we were measuring. We were just saying that's the gas that makes bread rise. So we were measuring with our ruler how much gas was in each tube, and that's how come glucose got first place and fructose got second. But you have to know what gas we were measuring. It's CO2. CO2. You're okay on question number 4 on the fermentation tubes. Okay, question number 5 did that. Aerobic respiration experiment. Okay. This was a day of big fun. Here's one of the setups that we used. There was shaving cream involved here also. I don't know if you remember. This was the shave. Some of you remember the shaving cream more than others. Okay. Yes. And you may remember tube number 1 had some germinating peas. Tube number 2 had dry peas with glass beads. And tube number 3 was all glass beads. I want to direct you to page 79 to take a look at that. This is on page 79. You guys remember this? Okay. So, which direction did you expect the shaving cream in the pipette? And you had to put it inside this pipette right here. In case you don't remember, we stuck the shaving cream here, squeezed the button just a little bit, and put a little bit of shaving cream in. That was the biggest thing, or we tried to, yes. And we marked it, and then we did some other things, and we came back later to see if the shaving cream moved. Okay. Which direction did we expect the shaving cream to move? Did we expect it to move in, or did we expect it to move out? In. It moved in. And the reason we expected it to move in is that the respiring cells created a vacuum. And if you don't like vacuum, which I think is spelled this way, did I get that right? Is that how you spell vacuum? You could use the word suction, if you like that better. That makes more sense to you. So then that begs the question, why did they create a suction? They created a suction because they were using up some gas. Some gas was being used up in the test tube. What was the identity of that some gas? Oxygen. Because they were respiring aerobically. This one was aerobic cellular respiration. Now guys, on this test tube, you can see I put aluminum foil on it, but if you're close to the front, you can see I left the aluminum foil open in the front just so you could see what was inside of it. But the day we did it, the aluminum foil went all the way around. Okay? Why did we put aluminum foil on these? Make it dark. Why did we want it dark? Yeah, we didn't want these little peas to get any light because what would they do with that light? Photosynthesis, which was a competing reaction. So we were trying to keep them from doing photosynthesis and only doing respiration. Okay. Okay, look over cellular respiration 1 through 7. Make sure you're okay on that. Everybody okay on that? Great. Okay, you have to know the stages of mitosis. You've got that great handout that you did for me for homework and got some extra credit on that, so I'm going to breeze past that. Here's the cell cycle. It's on page 86. Page 86. My picture is just a little bit different than yours because I took this picture from your textbook, but it's pretty much the same thing. I do want to tell you that we are not responsible for prometaphase, so if you didn't mark out prometaphase, make sure you mark that out. Okay, I have know the form of DNA at each stage. So I want to talk to you just a little bit about that. What's the difference between chromatin and chromosomes? Okay, one of them's real spread out and one of them's tightly coiled. Which one's real spread out? Chromatin. So it'd be kind of like this. Whereas a chromosome would be like this.
Just because of 2 instead of 1.
Okay, I just drew, you're right, I drew my chromosome and chromatin so far single-sided. Now, I want you to look on page 86. Where in the cell cycle would a single-sided chromosome become double-sided? Page 86. Look at the picture at the top of the page. Benjamin is nominating S. Do you agree? Why? What happens in S? The DNA is replicated or duplicated. So you're right. Then it becomes double-sided. And it looks more like what we're used to seeing. Okay, so I want to back up to the cell cycle.